Good afternoon. I'm Merage Jacobson. I'm the director of the Center for Healthcare Management and Policy at the Paul Merage School. Thank you for joining us for our spring speaker event. This is the first of what I hope will be many collaborations with the Center for Digital Transformation. What I'm so excited about is that both this speaker series and the speaker who will be introduced shortly and the mixing of audiences is that I think this is really what's going to be crucial to unleashing the power of kind of big data, precision medicine. That is to say, I think we need more than just the technology, the data. We need physicians or clinicians of all types, technologists and policymakers all at the table together. So I'm really pleased that we were able to draw such a group today. With that, I'm going to turn it over to my esteemed colleague, Professor Vijay Gurbaxani, who is the Taco Bell Chair of Business and Computer Science at UCI, as well as the Director of the Center for Digital Transformation. So thank you so much.
Thanks, Mireille. I just want to assure everybody that's about the only time in my life I've ever heard her being so nice to me, so thank you. I run the Center for Digital Transformation, and before I get into introducing Atul, I just want to say a couple of things. One is this is a perfectly opportune time to say that our business school has repositioned itself as developing leaders for a digitally driven world. And what we mean by that is digital is transforming everything. And healthcare obviously is 20% of the economy, and that is one of the biggest pieces of the economy that are being transformed. We ran a 2-day program that's concluding today actually on big data and analytics. Some of the people who are there are here today. But it really speaks to what digital technologies make possible. So we're really sort of able to drive efficiency. We're able to discover new business models, often related to big data. So when Mireille and I started talking about, and I think healthcare is one of the sectors that everybody in this room, even if you are in healthcare, would agree needs to be fixed. It costs too much. One of the things that I always like to say about the healthcare economy, which frustrates a lot of my healthcare friends, is that it's one of the few sectors where nobody's happy. Patients aren't happy, payers aren't happy, physicians aren't happy. Usually there's at least somebody who's making money off the thing who's happy. Like, how can you have an entire sector where not very many people are happy? And every time I get sort of dismayed by what's going on, I just look to sort of people like Atul, because these are the folks who are developing sort of the next big wave of what is going to happen in medicine. And it's sort of the entrepreneurs who mix academic medicine, technology, and business, who are really going to, I think, help this country move forward in really exciting ways. So with that, I'm delighted to welcome Atul Butte. He is, and this is one of the things I think we're going to see more of, has a degree in computer science from Brown, and then has degrees in medicine from Harvard and MIT. He's actually worked as a software engineer at Apple and Microsoft as well, written for publications like Wired, has spoken at TED, and has launched some amazing big data-driven companies. And if you think about the world of sort of data-driven X, whatever that X is, this is a great example. Today we're interested in health, but I think the future is data-driven X, where X could be any industry that you can think about. With that, I'm going to get out of the way. And Atul, thank you so much for taking the time to fly down here and take it away.
Great. Great. Thanks for having me here. It's always great to be able to help out any of my fellow colleagues at any of the University of California campuses. And I love coming down here. This is one of the few day trips you can do from San Francisco. Everything else is far for us. But it's great to be here, and thanks for all of you to spend some of your time here today. First, I'm a medical doctor, so I usually have to start with my conflicts of interest, so I have just a few. So suffice it to say, I've started a couple companies. I consult for a bunch of companies. You might not want to believe another word I say over the next 40 minutes. I wouldn't blame you. But I'm most proud of the bottom right. Those are all the startup companies started by my students. More than half my graduate students now start companies, and they do this with the most amazing platform in the world. It's simply data. It's usually big data. It's often open big data. So I'm going to show you how they do it. I'm going to show you how I do it, and maybe I'll convince you this is the most amazing time to be in biomedical research and innovation. So a lot of physicians have to use a slide like this as a slide of shame. This is my slide of significance. In my lab, we do believe if you want to change the world, you can't just keep writing papers about it. You actually have to file some IP, and if there's no one wants to license that IP, it's up to you to start the company yourself. So we kind of get it in my lab. So with that, I'll start. If you haven't heard, by now, we're in this data deluge. My favorite example of this is this guy collecting ones and zeros with the umbrella. This is the cover of The Economist magazine almost 5 years ago now. You know, it's raining ones and zeros. But this is the issue where they announced that the human species generates 2 zettabytes of data every year. 2 zettabytes. Now, if you forget your metric prefixes, it goes kilo, mega, gigabyte, terabyte, petabyte, exabyte, zettabyte. Of course, the following year, it's 4 zettabytes, and 8 zettabytes, and 16 zettabytes of data, right? Because this is doubling every year or two. I'll be the first to admit, most of these zettabytes of data are these YouTube videos of kittens playing with pianos and yarn, right? Entertainment value, perhaps no scientific value, but there are scientific data sets in the zettabytes. So for example, the Large Hadron Collider in Europe generated petabytes of data for those scientists to find new subatomic particles, and in fact, that data is open to the public. But my favorite example is NASA. So NASA announced about 2 years ago that with the new James Webb Telescope and many other telescopes coming online, NASA is thinking by the end of this decade, which is not that far away, by the end of this decade, they might be able to generate an exabyte of data every day in terms of pictures, okay? An exabyte of data every day. Now, something interesting already about NASA's exabytes. NASA already admits that they generate so many pictures of the sky, there are not enough astronomers on this planet to look at all the pictures. And so how do you know they admit this, right? Because they set up something called Galaxy Zoo. You can go to Galaxy Zoo and take a test, and you become the one who assigns that spot in the sky. Is it a star? Is it a galaxy? Right? So NASA already admits they have so much scientific data, there are not enough scientists, traditional scientists, to look at that, so they have to crowdsource it and get citizen science, a new audience, to help them scientifically interpret this data. It's a different world now. And of course I'm going to tell you we're there in health and life science as well. Of course I'm going to tell you that. But you don't have enough astronomers now to even look at all the data you're generating. Now, in fact, we have so much data. This article came out in Wired magazine, not the most scientific of journals, but this was very influential to me. We have so much data that some people think science itself is getting obsolete, okay? Now, how do you say science is getting obsolete? That's kind of ridiculous, right? It's actually a scientific method. So Chris Anderson is editor-in-chief of Wired. He was back in 2008 when he wrote this article. A scientific method. You remember what the scientific method is? You're taught it in 3rd and 4th grade. You come up with a question, you call it a hypothesis, and then you go make measurements to answer the question, right? Here, I had a question, I'm going to go gather data, right? In 4th grade, it's like plants growing and stuff like that, right? But in today's world, we already have the measurements. 99.9% of the hard part now is figuring out what's the question I want to ask. Given this data, what's the killer question here that everyone's been dying to know the answer to, and no one's realized you can ask and answer that today, right? Given this data. It's not, the hard part isn't programming, coding, cloud computing, none of that. The hardest part is figuring out what's the question you want to ask, because you got a lot of data right now. What do I want to do with this data? What's the killer thing here? It takes a lot of inspiration, a lot of domain knowledge, and a lot of recognition of what the unmet need is in a field, right? That's hard for programming people to know. They just don't know that, especially in medicine. And we're certainly there in medicine because of devices like this. Now, I could have showed you any of the amazing gadgets we have in biomedicine. I could have shown you a sequencer with blinky lights. I could show you a mass spectrometer. I could have shown you a proteomic setup. I love showing these gene chips because this is how I got my start about 18 years ago in a lab in Boston. These chips were priceless. You put a sample of tumors or cancers. We had cancers back then. And you get a readout of every gene in the genome. So genes are like the recipes in our cells. There's 20,000 of them. And you get an idea of how much of each recipe they're cooking. More of this one, less of that one. They were priceless. But now they're so cheap, I carry one in every suit. And this is what they actually look like. I have one in my hands here, right? A couple $100, and you get a readout of just every gene in the genome. And everyone started using these. In fact, you very quickly get tired of measuring them one by one. And then you start to use the 96-well plate format. And that's so cheap, I can carry that in my bag. And I have that in my hand here, right? You see a picture of it up there. And then you get tired of measuring them 96 at a time. And now there's a 384-well plate format. And that's so cheap, I can carry that in my bag. I cannot more clearly illustrate exponential growth in biomedical data than to show you these plates, right? When I started, we had that one at a time, then this, and then this. We don't even use this one anymore, right? We've moved on to something called RNA-seq, where we use sequencers to do this. In fact, this company doesn't even exist anymore. Affymetrix just got acquired by Thermo or someone last year, right? This is how fast we're just making measurements. Here's a cancer, here's a biopsy, here's a cell line, here's a mouse model. Let's go make some measurements, let's go write a paper about it. And what happened is, in early 2000, 2001, 2002, everyone started using this particular type of technology. And all the journals, the scientific journals, they started getting inundated with these papers. Here's a list of genes that change in cancer, or that cancer, or heart failure, diabetes. And you know what the journals did? The journals said, they took a look at this, they said, you know what, we're getting too many of these papers with these gene chips. No more papers with these gene chips. That's it, okay? That's enough. Unless you put this data into an international repository where all the reviewers can double check all the math, probably important to do that, we're getting closer to the clinic, and maybe scientists can use these samples for something else, not just this paper that you're submitting. The precedent for this in science goes back a long time, almost 45 years ago, to something called GenBank. Most people have never heard of GenBank. Maybe if you look at biomedical papers, you know PubMed, it's the next door neighbor there on those tabs at the top. GenBank goes back to the very first papers at a DNA sequence, okay, AGTC. Nobody wants to type in that again, right? They're seeing it in a paper. They were sending tapes to each other for 45 years. Anyone remember 3.5-inch discs, right? Remember 5-inch discs? Remember 8-inch discs, right? Before that were tapes, right? They were sending tapes to each other 45 years ago. Of course, it's all on the World Wide Web, but the precedent has been set. If enough scientists use a molecular modality or measurement modality, you got to share the raw data at some point. So again, I could have showed you anything about them. We're going to focus on these kind of chips for a second. So fast forward to August of 2012, when this paper came out in Nature, featured my labs and many others, we hit this milestone of 1 million samples publicly available. 1 million samples, someone's biopsy, someone's cancer from the operating room, someone's cell lines in an experiment, someone's mouse model. De-identified, I can't figure out who these people are, but if you want to know which of those genes, which of those recipes are being cooked to that cancer or this diabetes, 1 million samples publicly available. Now here's the growth, right? I started in the field here at 0, here's 1 million, right? Exponential growth. Now if you look really carefully, doesn't it look like it's slowing down there at the end? Kind of looks sad. Then you realize they only counted half that year, right? This came out in August of 2012. Right? We're still doubling about every 4 years. In fact, we just hit 2 million samples publicly available. Now, what does this mean, 2 million samples here, right? Any scientist and any pharmaceutical company, any college, any university who wants to study a disease, instead of going to get ethics and IRB approval and go getting biopsies and go getting the pipetter, you start with data now because probably someone's measured it already, right, for the past 20 years. So you start with downloading data instead of actually going to recruit patients. Oh, forget about your scientist and my scientist. Even a high school kid, a high school child, let's say who needs to do a science fair project, right? Let's say she wants to do a science fair project on breast cancer. She can go to these websites, type in breast cancer, and hit search, and find and download 76,000 samples of breast cancer, about as easily as she can find a song on iTunes today, right? 76,000 samples of breast cancer. Now, what's so magical about 76,000 samples of breast cancer? If you haven't figured it out yet, that's more samples of breast cancer this high school kid can get to than any breast cancer researcher will ever have in their lab. Because every one of them eventually has to submit data into the middle, into the database. And whoever figures out the database now has more data than any investigator in the field, any researcher. Maybe it's not breast cancer you care about, colon cancer, prostate cancer, diabetes. Probably someone's already studied a disease of interest, and the data is just sitting there waiting for you. Because what do most researchers do when they go make these measurements? They go get a big grant from NIH or NSF. They go make some measurements, they go write a paper, and what do they do next? They go get another grant to go make more measurements and submit more data, right? Very few people trained to use the data already sitting there, right? It takes a different kind of investigative skill. It takes a different set of experiences, more computational people, right? It's like everyone knows how to upload, very few people have been taught how to download, right? Think of it as a bank where everyone's been taught how to deposit, no one's been taught how to withdraw. Hell, wouldn't you want to learn how to steal money from that bank? It's just sitting there, right? That's what this is. The data's just sitting there. Now, actually, the number I like is this number, 3238. Let's call that 3,200. 3,200 research labs and research groups have already submitted data on breast cancer, roughly about 3,200. Let me tell you what a ridiculous number that is. Let's pretend I'm a brand new researcher in breast cancer. Okay? I'm a newbie. I know nothing. And I'm going to write a grant for those of you who are academic, or I'm going to write a business plan for the rest of you who are more business-oriented. Let's pretend I'm writing a grant or business plan. In fact, those two are quite similar nowadays. Let's say I'm writing a grant or a business plan to study breast cancer. And in my grant or my business plan, I propose to you, you're going to evaluate my grant or business plan, I propose to you that I can get 3,200 of the best breast cancer labs in the world to get their best characterized patients, run the latest measurement technologies, and I propose to you, I know a way to get 3,200 of those labs to share data with me for free. For free. Some of you are already chuckling. You would laugh me out of the room if I wrote this in a grant or a business plan. Why would 3,200 of the best labs in the world share any data with a newbie for free? Yet here it is, just sitting there waiting for you. So when I think of public big data like this, the phrase I like to use is retroactive crowdsourcing. Okay? What does that mean? These 3,200 labs are there to help you, and they don't even know they've been helping you, right? Actually, these 3,200 labs are there to help you, and you don't even know they're helping you, right? And I don't know, if 76,000 samples are still not enough for you, just wait another couple months, it'll be 100,000 samples or 200,000 samples, right? In this hour, we will add 120 samples, just sitting here doing nothing. Someone in the world is uploading data into the system. And all that data is just sitting there waiting for you. Now, if you have any doubts a high school kid can do this, here's the high school child that did this, okay? We were just talking about high school kids, right? This is Brittany Wenger from Sarasota, Florida. I've yet to meet her. I talk about her in all my talks. At 17 years of age, she won the top Google Science Fair competition, first prize, because she built an artificial brain to diagnose breast cancer. This was in 2012, so it's been a while. I think she's a Duke undergrad now. She's probably in grad school at this point. Artificial brain, so deep learning neural networks. She downloaded off the internet immunohistopathology images and qPCR data. She built a diagnostic. I guess she was bored because the following year she did this for leukemia, so she was in the paper all over again, right? Of course, what I mean is if a high school kid can do this, we all better learn how to do this now, right? That's what even a high school kid can do this, right? We all got to get there. It's amazing. We just don't have many people trained this way, right? Well, that's what high school kids want to do with this data. What do we want to do with this is get to precision medicine, right? So what is precision medicine? Well, President Obama mentions precision medicine. It's a big deal. Precision medicine has a lot of different definitions. But what I want to do, what I think about precision medicine is I want to customize the medical care we're going to deliver to a patient. We're going to customize the medical care. And how are we going to customize it? We're going to base it on the measurements we're going to get from those patients. They might be biological measurements, like your DNA or proteins. It could be your behavioral measurements. Maybe you like certain restaurants more than others. It might be your preferences. I might get some of that from your smartphone, your wearables, your apps, right? But actually, it can't just be about your measurements. I need a lot of those kind of measurements on a lot of people. And even more importantly, I need to know what medical care did I give to the rest of the population and what's working and not working, and then apply it to you, right? So it can't just be personalized. I can't just do it with your measurements. I need to have a great reference library, a repository of what's working and not working in medicine and apply it to you, right? So that's what precision medicine is. Now, a lot of people have different definitions. Obviously, a lot of data-driven problems there. But for me, there's three fundamental problems, challenges that we have in precision medicine. First one is coming up with better diagnostics. How do we know if a patient has a disease? We need better therapeutics. We're going to need to have better ways to treat them, especially with drugs. And then the third one is figuring out what are we doing to the rest of the population and what's working and not working. Getting a handle of the medical care we're delivering. That's the third challenge. So all I'm going to do for the rest of the, 35 minutes or so, is I'm just going to give you our stories of how we're coming up with better diagnostics, better therapeutics, and figuring out what are we doing in medicine right now that's working and not working. Coming up with just anecdotes for you here. All right, so first story is coming with diagnostics. What's a diagnostic? A diagnostic is a way for me to tell, as a physician, does a patient have a disease or not, right? I might have imaging diagnostics, like X-rays or CAT scans or MRIs. I love protein tests, and I love blood tests, where I can tell with a protein, does a patient have a disease or not. Now, to explain what proteins are, I'm not going to go into proteomics and mass specs. I'm going to give you a simple example that everyone knows, right? When I was a pediatrician, we had this all the time, where a toddler would come in with a sore throat and a fever, right? And you're trying to figure out, is this strep throat or not, right? Now, you've either had this done or your children have this done. We can stick this Q-tip back there somehow, right? Put some drops on this thing and tell if it's strep throat or not, right? That's called a rapid strep test, and that's actually done with something called an ELISA, enzyme-linked immunosorbent assay. It's 1970s technology, okay? But it works in a desert. You don't even need a refrigerator for those things. I love those kind of tests, right? Not fancy RNA and stuff like that. Those work. You can get decent value in a company that way, but those are the cost of goods there are super expensive compared to ELISAs. I love those. So my first big grant from NIH was, we have these repositories. We have all this data. Let's just start by getting every single disease that's ever been studied with these chips. Just go get them all. Why even pick this cancer or diabetes? Just go get all of them. So we start to pick all these kinds of experiments where in the same experiment, someone studied diseased individuals and healthy in the same experiment, okay? Disease and healthy, disease and healthy. And each box here is a gene or one of these recipes, and the colors mean more of it or less, and you calculate signatures, right? What's changing from healthy to disease? And Marina did a lot of this early work, and now she's a faculty member of her own. She helped make these slides. In fact, when you pick any disease, if you pick every disease, some diseases you have thousands of people studying, right? I showed you breast cancer. So many people just studying breast cancer again and again and again, and we can start to put all of these together to make diagnostics. Now, it's always dangerous to show a little bit of biology here, but we are kind of in a biology building here, so we should show some biology here. And just get into the little technical details here for a second. There's DNA, RNA, and proteins. And this is what I love to get to, right? When you stick that Q-tip in, we want to measure these kinds of things. But these chips give us the blueprints of these RNA, right? That's what we measure with these chips. And so obviously, if the RNA is changing, maybe the protein is changing, so we're going to just use this kind of arrow to make this kind of leap of logic. So we started to make a bunch of diagnostics. We made cancer markers. I'm not going to spend a lot of time talking about these. This is one where the pediatric brain cancer surgeons, that's a depressing field to be in. The pediatric brain cancer surgeon said, well, sometimes we see these kids with brain cancer, medulloblastoma, and they can't tell where's the cancer and where's the brain here, right? And you want to keep as much brain as you can in a child, obviously. So they came to us and they said, well, can you design some kind of paint? Just paint. Like, just put some paint on the operative field. Maybe we could turn off the lights and maybe it'll glow where the cancer is. How the hell do you design a paint like that, right? How do you do that? Well, you start by downloading every public data set you have on pediatric brain cancers, medulloblastoma, and then you download the normal neurons, normal astrocytes, normal glial cells, everything else that's normal there, and digitally subtract those. And then you got a bunch of genes that are up in the cancer and not in the normal cells, and then you figure out which ones might be on the cell surface that a paint could hit them. And when you do all that, here are two of them where it's darker here than here. That's a paint, basically. So we can do that. And then we came up with a blood test for transplant rejection. To make a long story short, we can do organ transplants now, kidneys, livers, lungs. Sometimes patients wake up and the body says, this is not part of me. I have to get rid of this organ. That's called acute rejection. It's not painful. Patients don't know this is happening. So to tell if this is happening, we stick in these long needles every once in a while to see if there's rejection going on. We said we wanted to come up with a blood test, a protein blood test for this. And how do you do this? Well, download every rejection data set there is, do a big Venn diagram. What do they all see in common? And we came up with a universal blood test. Whether it was kidney, liver, or heart, the same markers we designed to go up. So it didn't matter which organ you put in. If it's rejecting, we got these markers as well. So I won't spend any more time talking about these two. I will spend a little bit more time talking about this disease, preeclampsia. Show of hands if you've heard of this condition, preeclampsia. So it's about a third of this audience. It's usually a third of the audience. This is a third of the audience that watches Downton Abbey because one of the characters dies of this disease. Spoiler alert if you haven't seen the series, okay? Eclampsia is when you're pregnant and you start to seize, your blood pressure goes shooting high. This is still how husbands lose wives, so preeclampsia is a little bit earlier than that. And this disease falls right between the cracks in medicine. So the obstetricians, I love them, and I have great friends who are obstetricians, But they come to this and they say, boy, blood pressure is going up. This is a danger to the pregnant woman. Let's just take out the baby. The delivery is the cure. Just take the baby out. The mother will be fine with some mag sulfate and stuff in 48 hours. And then we pediatricians have to keep these tiny little babies alive for months, right? Because they're born very early. So it's right between the cracks of those two fields. 20 billion a year in the US spent on this disease. This is still how husbands lose wives around the world. We have 4 drugs in trials for preeclampsia in the United States. But the diagnostic is ancient. Okay, we look for a protein in the urine. One of the most nonspecific tests that we have in today's obese America, where a lot of people have protein in the urine now, right? It's not even a big deal. So we wanted a more specific blood test for preeclampsia. So how do you do this? Well, Linda is a grad student who got started. Bruce did the protein work later, and Matt got involved in a moment, you'll see. You go to the websites and you just type in preeclampsia, preterm birth, and you have 266 experiments you can download. Now, it's not 3,000 like breast cancer, but hundreds of people have already studied preeclampsia using these kinds of chips, right? Here are 94 samples here. There's 249 samples here. Here's 104 human samples here. Some are in 2012, 2009. And how does this work? You just download all of them. And you do a big Venn diagram. You know what I mean by Venn diagram? All those circles that intersect? I'm looking at what's in common here. What do they all see in common? You know, it's really funny in my way of doing science. I love these people who submit their data. I don't trust any one of those experiments, but I trust what I see in common, right? If a whole bunch of them are seeing the same markers up or down, I'm going to go chase those down. So we get a bunch of ideas. Here's a dozen. Here are 24 markers that we could make into blood tests. And then we get stuck. How do we get stuck? Because we as computational folks, we could turn our crank and go like crazy and come up with ideas. But we got to test them in samples. And how do you get those samples? Well, you could go slow down for a second, go to the medical system, go get your ethics board approval, go put up those posters. You've seen the posters in the hospitals, right? Go join my study. More often than not today, instead, we just Google for it. And we find companies like this, Prometics, Specimen Bank, human clinical samples and patient samples, all IRB approved, subject had informed consent. Okay, here's our spring 2011 biorepository, pregnancy collections, 2,000 samples. What would you like to buy? First, second, or third trimester, high risk or low risk of preeclampsia, 15 milliliters each, $45 per sample. And we bought them all. And on the protein side, you just buy off-the-shelf ELISA kits from these companies. These are the ones we used. What is this, by the way? What the hell is this, is what you're wondering right now? Turns out when you're a hospital, when a doctor orders a blood test, right, they run the blood test, but they save the blood there for about 48 hours in case the doctor wants to add something else to the test. At the end of 48 hours, they got to make room in the fridge. They got to throw out that sample. And something magically legal happens on the way to the trash. It becomes a discarded human sample. If you strip off the sticker, put on a new sticker, de-identify the samples, companies actually come in and help you organize all this, and you can use this for research use today. Now, these patients are beyond it. This is actually consented. But that's what you can do. And companies come in and sell this for research use. And these show up in 72 hours on dry ice. This is not the hard part anymore. And so we get the samples, buy the off-the-shelf kits. Here's an example, one marker that works, hemopexin. It's up in preeclampsia compared to normal, healthy, pregnant women, okay? You can just see the pink bar is higher than the green bar. That's all I'm trying to show you here. There's p-values for that. So we got Marchedime's money to start. We had an internal seed grant called Spark when I was at Stanford. We wrote the papers on that. You can find it all at that link. And what do you do next in Silicon Valley? You start a company on this. Right? So here's the arrow. You can see a couple of these arrows. You start with the unmet need. We don't write a line of computer code unless we know what the question is we're going after. We want a diagnostic for preeclampsia. Public data available. Marchedime's funds the work, especially Linda. Data is analyzed. We design the diagnostic. We get Spark grant, $50,000. We buy samples off the internet. We then also validate in other repositories at Stanford, UCSF. Spin out the company, more than $2 million seed financing. Now let me be crystal clear here for a second. I'm not showing you the $2 million here to brag about it. It's actually not a lot of money to raise in Silicon Valley, as you know, okay? I am showing you $2 million here to try to convince you this is the new way science has to continue out of academia. We know we can get this test to work in samples from the freezer. The next science experiment that needs to happen is to test this in a prospective multi-center trial. Does this work moving forward now? And that scientific study is now going to be done in the startup company funded by private dollars. Or to put it another way, do you know how hard it is to get a brand new $2 million NIH grant today? The science experiment continues in the startup company funded by private dollars because it's relevant to patients now, right? If this works, you're going full steam ahead. That's what this means by starting companies. Now, I love this story. You can find the whole story with the links here. I love this story because we already sold the company. It's already been acquired by Progenity in San Diego. So we went from public data, which people think is as valueless as kitten videos, to testing, developing this, testing the samples off the shelf, launching the company's $2 million seed financing, selling the company, start to finish, 24 months. Inventors happy, investors happy. Of course, we're going to do more of these, and I'm giving away my secrets here, right? Because every one of you could pick a different disease, and we'd never step on each other's toes. Such is the need for diagnostics in medicine today. That's how many we need today, right? Literally, start to finish, 24 months here. We've already exited the company. That's the first diagnostic. Let's talk about the second story here. The next big diagnostic is going to be DNA sequencing. Now, if you haven't heard, we're in this major revolution that's getting cheaper and cheaper to sequence your DNA, right? I showed you the DNA, RNA, protein. That DNA, boy, you've heard of companies like Illumina and many others. Illumina is the market leader. The DNA sequence, so easy numbers to remember, it's about 3 billion base pairs, 3 billion letters, 3 billion from your mom, 3 billion from your dad, and costs $3 billion. The Human Genome Project took $3 billion, 13 years. It was done in factory-like settings like this is the Whitehead Institute in Cambridge, Massachusetts. And now, literally, we're heading to USB stick sequencers. It's a company called Oxford Nanopore. This is my hands holding this. That's literally a USB port that powers this sequencer. It is a no-brainer that DNA sequencing is going to be a major revolution in sequencing and diagnostics. No-brainer. It's coming. It's already here, as I'll show you. So that's the second diagnostic I'll talk about, DNA sequencing. Now, I'm not a geneticist, but we got involved, my lab got involved in a very unusual study while we were at Stanford. This patient here, Steve Quake, was the first patient to show up with his genome sequence. Now, he's a very unusual patient. He's a bioengineering faculty member at Stanford. He starts sequencing companies on the side. He started a company called Helicos. And he had published his genome sequence. I think he was the first faculty to have it sequenced or whatever in 2008 or so. And 2009, he showed up as a patient. Because here's Steve Quake in the family tree here. His nephew down here just died at 19 years of age. Sudden death. Did not wake up one morning. Sudden cardiac death here. So here's Steve. Here's the nephew. And so Steve Quake goes to his cardiologist, Ewan Ashley, and says, "Doctor, am I going to die of sudden death? And doctor, here's my genome," right? So this exercise, a team of about 100 people or so, this became an exercise of how is a patient and a doctor going to interrelate over their genome in the future. This paper came out in 2010. It was one of the most highly cited papers in Lancet in all of medicine that year. And so this became a very amazing team effort. First, let's talk about the heart. So Ewan Ashley spent a year looking at the heart and all the heart genes. It turns out Steve Quake does not have any known lethal variants in his genes that sets him up for arrhythmia. So that's good. Okay, that's off the table. Russ Altman said, well, since you have a genome, let's look at all the drugs that are out there, pharmacogenomics. 150 drugs we can say something about. Would the drug work? Might there be side effects? What the dose should be, like blood thinners, like warfarin and statins. We can look at all those side effects, 150 drugs, and his team worked on all the drugs here. So heart was done, the drugs were done, and all the rest of medicine came to my lab, okay? Evaluate every other disease that's out there in medicine. So how the hell are we gonna do this one, right? So this was in 2009. In 2007, 2 years earlier, we had this idea that someone was going to show up with their genome at some point. 23andMe was just launched. We knew this was going to happen. I was looking around in 2007. I realized there's no master list of just every spot in the DNA and every disease, right? If you're going to have a genome in front of you, you kind of need a table, a reference table. This misspelling leads to this disease, and this one leads to that, and you need all the numbers behind it. And nobody had that master list. So we started just that summer by hiring a high school kid from San Jose High School. We gave him 50 genetics papers to read. We say, we want you to get every fact you can out of these 50 papers, right? All those spots in the DNA, the SNPs is what they're called, and write down every disease, every variant, every number that they have in the paper. We want you to curate these 50 papers. And he was able to do it. He got 50 papers done in about a summer. Smart high school kid, right? So when we start to realize, all of those genome differences, these misspellings were locked up in papers. We had to manually read every one of these papers. I love natural language processing. I'm a computer scientist. It doesn't work here for a lot of reasons. I could spend an hour telling you why. I'm not going to spend an hour here, though, right? Then we extrapolated. Well, if a high school kid can do 50 papers, how long would it just take to read the entire field of genetics? And that's what we did. So here's one paper, here's 6 papers, here's 100 papers, here's 1,000 papers. I literally have a kind of Perl script that makes these kind of graphics from each PDF. We can do this. And we've blown past 20,000 papers. And we basically read the entire literature in human disease genetics. And because I run my lab like a startup company, I outsourced all of this to a team in India, 100 geneticists and bioinformaticians. I couldn't even find the space in Palo Alto to even house 100 people. It's impossible. But I didn't care about the curation part. I wanted to work with the list once we had the list. That's the more important part. That's the valuable part. So we got a team of geneticists to just read every single paper. We have the master list. And what do we do? Well, we start to use that now to look at every disease. So this is literally what we gave to Steve Quake, a patient. This is an info viz we came up with. Each font here, each word here is a different disease. And the font sizes are bigger for the disease that he's higher risk for, right? So coronary disease, like his parents have had. He has a lot of coronary disease in his family history. Type 2 diabetes, arthritis. And what we also paired with that around the edge were all the known behavioral and environmental factors that influence if you get those diseases. To me, this is the prescription pad of the future, right? What can I do to compensate for my genome, right? What can I do to compensate? So these are all, we mined the heck out of PubMed literature, and we came up with, well, Steve Quake, you should watch your smoking, you should exercise, you should watch your diet, you should probably watch your alcohol. Probably don't need a genome sequence for those 4 pieces of advice. I grant you that, I know, right? But in tiny font at the top is pesticides and Parkinson's disease. And he's at slightly higher risk for Parkinson's disease. And there are 10 papers in the literature that have linked pesticides to the etiology of Parkinson's. I don't write the papers. We find them in the literature. So we said, Steve, if you like to hang out in the garden, you should stay away from pesticides here, right? I think this is how patients and doctors can talk about their genomes in the future, not which base pair and AGTCs and all that stuff. You're doing great with stress. Come back in 6 months, work on the smoking. Okay, you're back. All the risks have changed, right? Because you're getting older. You're not going to get all that childhood stuff. You're going to get adult stuff. This is how we can think about talking about your genome based on what you might get and what you can do to compensate for it. That's one way to think about genomes in healthy individuals. Of course, genomes are used in many other ways, including this whole new field of rare children, so children with rare syndromes. It used to be you need like 100 kids with a rare syndrome to write up a syndrome, and then it was down to 10, and now it's down to 1. I'm not going to spend any time talking about Nick Volker. This is a Pulitzer Prize-winning story. This is literally the first child saved by DNA sequencing. Pulitzer Prize-winning story. When Francis Collins, director of NIH, goes to Congress, this is literally the poster child he brings with him to Congress, showing how this child was going to die in the ICU at a pediatric hospital in Milwaukee. The sequencing saved his life because based on that, they did a stem cell transplant, and he's out of the hospital. He goes to school. He's not out of the woods, but they saved his life based on DNA sequencing. And you might know, that cancers are done. Foundation Personalis, many other companies now sequence cancers to try to figure out new therapies because we have a lot of different cancer-targeted therapies now, and neurological diseases are becoming routine. And what do you do next in Silicon Valley? You start a company on this. So we started Personalis. 4 faculty founders, and John West, who's the CEO, he ran a company called Solexa, sold it to Illumina. That is the sequencing that they do now. So you start with the need to use genomes to predict disease. We got the publication security. We had donor money to get this launched. Launched the company, and we've raised more than $75 million from investors now, past Series C. 150 people in the company based in Menlo Park. We came out of stealth mode by getting the Million Veteran Program contract. So we're sequencing those in that research study. And to put it another way, do you know how hard it is to get a $75 million NIH grant today? The science continues in a startup company. We literally started with 50 papers and a high school kid back in 2007. Let's switch to therapeutics. Much worse of a story. Therapeutics. We're going to have a problem with therapeutics. Why? Because it's costing a ridiculous amount of money to develop every new drug. Right? So if I were to ask you how much does it cost to develop a drug, the answer most people know is it takes $1 billion, it takes 10 years to develop a drug. Right? $1 billion in 10 years. A lot of people know that number. But $1 billion is an underestimate. It's an underestimate. How do you get the right number? Well, Matthew Herper does this in Forbes every other year or so. It's not complicated math. I know it's a little bit after lunch. Let me give you some math here. Here's the formula. How much should you spend on R&D divided by how many drugs did you get, right? Simple math. How much do you spend divided by the number of drugs? If you do that math, like Matthew Herper does, for these top 12, it costs between $4 billion and $12 billion per drug. Kind of not sustainable. Just not going to be sustainable. Now, actually, that's a really negative way to paint the pharma industry. Let's paint it a different way. Even if every pharma and biotech company on the planet was 100% successful in developing the drugs they're trying to develop, there are not enough of those companies on the planet to develop all the drugs we need in precision medicine. It's the whole NASA astronomer problem all over again. Even if they all knew what they were doing, there are not enough companies, right? There's just not enough of them. We need to think about this a very different way. We cannot keep believing it has to cost billions of dollars to do this. Otherwise, we're sunk. We're just not going to have new drugs now. So we have to come up with new ways. And of course, I'm going to try to come up with ways using public data. I'm not going to say that we've solved the whole drug discovery problem, but that's how we're going to go after this, right? And so as we're collecting all of these disease samples, disease and healthy, we see an enormous number of people depositing drug data onto the internet. Here's some cells in a petri dish with a drug, without a drug, with a drug, without a drug. So that's the Broad Institute and many others now just dumping drug data and researchers dumping disease data on the internet. And we're going to put these two together. The reporters for this call this Match.com for drugs, okay? Right, you know, Match.com, you find a spouse or maybe a date or something, right? A spouse or mate. And what's the famous saying if you're trying to find a spouse? Opposites attract. You've heard of that, right? Opposites attract. Same concept here. I'm going to show you all of the sophisticated bioinformatics with my arms. If I have a disease where this gene goes up and this gene goes down, and I can find a drug now that knows how to make this one go down and this one go up, maybe there's a match there, right? I'm just looking for drugs that reverse the effect of the disease. It's that naive. That's with 2 genes, imagine 20,000 genes. Right? It's that naive. I'm not trying to look for drugs that get to the cause of the disease. I'm not looking for drugs that might cure the disease. I just want naively want a treatment that might reverse what I'm seeing in the digital pathology here. All right, so we turn our crank. We're connecting drugs and diseases, disease and drugs and diseases. And we got a lot of ideas. But where we got a lot of interesting ideas aren't the new drugs. It's the new uses of the old drugs, drug repositioning, right? Now, we didn't invent this concept, drug repositioning, right? It goes way back, actually. A lot of people know the classic stories. The first one is an interesting cardiac drug, has hair growth as a side effect. Of course, that's minoxidil, right? Everyone knows the other cardiac drug that had its own interesting side effects. Nobody wants to return the drug after the trial. That, of course, is Viagra, right? Started as an angina drug. Actually, Viagra's done a double switch. There's another drug called Revatio, same molecule, that we use specifically for pediatric pulmonary hypertension. So that's when the blood vessels from the heart to the lungs in babies is too small. It kind of opens them up. Everyone was teasing about babies on Viagra at the time, you know, but it happens to work there too. Instead of finding these new uses by accident, how about we find them on purpose using public data? So that's a concept. So we turn our crank. And we got lots of ideas. This drug could work there, and this drug could work here. And then we get stuck. We're stuck again. Because how do you know it's actually going to work? Well, you gotta have to run a clinical trial, right? You got to put this into patients. Oh, hell, no one's going to put this into patients until you show that it works in an animal model. You got to show this works in a mouse model. So we go to all of our mouse friends, and some of them help us like crazy, and the rest go scattering off, right? Because they know we're going to generate a lot of work for them. We've got a lot of work with our friends and collaborators and core facilities. But now, when I really, really need to get a mouse study done, I go to assaydepot.com. Kind of sounds like Home Depot, and you'd be right. Let's go browsing. Here are my aisles: biology, chemistry. DMPK is drug metabolism pharmacokinetics. If you don't know what that stands for, don't click there. Pharmacology, toxicology. Right? Let's click on pharmacology here. What do you want to buy today? Bone, cardiovascular, dermatology, diabetes, general infectious disease, inflammation, neurology, cancer, eye, ear, pain, respiratory. What type of mouse do you want to buy? I don't know. I'm usually a diabetes guy, so let's click on diabetes for a second. Here, I can order a OB diabetes model, 16 mice. Okay, so maybe I have a drug and I want to put like drug and control, so 8 and 8. Or if I'm doing a dose ranging study, I might have 4 groups of 4, you know. If you don't know what OB means, if you read here, it says this mouse has been eating a lot and getting diabetes since 1951. It's a standard model of diabetes. It's neither the best nor the worst, but a lot of people use this one. And in 28 days, this company will get you the insulin tolerance test, the glucose tolerance test, the fasting blood sugars, and you'll know the data, whether this works. You send the drug blinded, you get the results back. Does this drug work or not? What am I covering up here? The price. $9,000 for this service, 9-week turnaround time, and I kid you not. Add to shopping cart. I can buy an entire mouse model off the internet with a credit card today. Now, what the hell is this, you're wondering, right? These are called contract research organizations, CROs. They've been around forever, especially all these little guys. Pharma uses them. But now because of Assay Depot, which is now renamed to Scientist.com, there's Science Exchange, there's pricing transparency, the price comes down. Even we in academia can afford this. Certainly startup companies can afford this. And it's not just about getting the mice done. I can work on diabetes 1 week and cancer the next week, right? I don't have to love my mice. I don't have to breed them. I don't have to do anything. I just get the experiment done. Now, some of you with sharp eyes are noticing this is done in China, okay? Some of you may have a problem with this, others not. I don't, but you're questioning the quality here. Actually, when you hit add to shopping cart, it presents you with 133 companies that want to do this for you, including Maryland and Wisconsin. So outsourcing doesn't always mean offshoring. You can pick wherever the hell you want this done, whichever country you want. You're still questioning the quality here. There's certifications down here. I can click here. If I need them to be FDA certified, click here. There's 5 of them, triple ALAC 28, ISO 9001, 7. You click on the certifications, you're only going to see the lists here of the certified ones, right? Some of you are still really, really concerned here. Because you know, when I get this to work, I'm going into humans next. How do you trust the results of this company? I'll simply say, if I really, really, really need to know if this drug is working in this mouse model, I just order 2 of them for 2 companies. Hell, at $9,000 each, why don't I order 10 of them from 10 companies? So why am I answering it like this? Think about a mouse researcher that gets a mouse experiment done today. Do they go across the hall to get a completely independent team of researchers to repeat the entire experiment? You know nobody does that. But I can hire as many independent teams as I want with my credit card. And now what I can do, in your mind, you had this problem with quality, I can now make it a function of dollars, right? If I have more dollars, I'm going to hire more companies. Not enough dollars, maybe I just get 1 or 2 of these, right? The quality problem now becomes a function of dollars. I just have to get the dollars. We typically will order 1 from the West Coast and 1 from the East Coast. I have nothing against flyover states. Probably should send more business there. But that's how we do this, right? That's how we get it done. I can outsource all of that. This is not the hard part anymore. 28 days, you get an answer back, right? So we've done a bunch of these, more than a dozen. This is one of my favorite ones. There's this computational prediction that imipramine, it's an old antidepressant, it's still in the pharmacy, would work on small cell lung cancer. Humans smoke to get small cell lung cancer. You knock out these 3 genes in a mouse, this mouse gets mouse lung cancer. So you see the nodules here. Takes a couple weeks. But without those 3 genes to protect the genome, this mouse gets mouse lung cancer. You can see all the people that helped us: Joel, Nadine, Julian, Alejandro, and Joel here. And this mouse has been treated with imipramine. Now, we don't use imipramine for depression anymore. We have much newer drugs, serotonin reuptake inhibitors like Prozac, right? Imipramine has some side effects. It can make you sleepy. And in some people, it might set you up for what's called QT prolongation, might set you up for an arrhythmia. Actually, neither of those 2 side effects sound as bad as having lung cancer, 5% survival rate in 5 years. And the cancer is melted away. It's gone in this mouse, completely gone. So we've done a dozen of these. Why am I showing you this one? Because we start with public data. We get this prediction that this drug is going to work. We test cell lines in a petri dish. I'm not going to show you that. We get this amazing lung picture. 15 months after the computer prediction, we get the ethics IRB approval. We launched a clinical trial on this one. Patients are being dosed with this. Total cost of that trial, $50,000. Not $1 million, not even $100 grand, $50,000 fits in a seed award. This is a phase 2 AAAAA study. Is there any biology at all here? If it works, we'll do more, right? We just got to see any biological effect at all here. $50,000. Now, I'm financially connected to the molecule, so I'm not part of the trial. I don't care if this works or doesn't work. We got to do so many more of these in academia. There is no company targeting small cell lung cancer today. There's no pharma or biotech. It's us or nobody at this point. And think about how many other diseases are there that are just waiting for someone to put attention on them. There's just that many. Here's one that just came out 2 weeks ago, gastroenterology. Here's an annoying disease called liver cancer. It's annoying because Americans actually don't get this, but 350,000 people around the world die every year. But not in America. Not a lot of people working on liver cancer. We have one questionable drug called sorafenib. This is a drug called nicotinamide. In fact, nicotinamide ethylenamide is the formulation, because you might have to change the formulation for it to work. This is liver cancer going out of control. This is a healthy liver again. Nicotinamide we used to use for worms, parasites, worms, okay? That's this drug. We computationally predicted that. You can see the glowing part is the cancer. You can see how it's better after treatment with NEN. In fact, we tested it in synergy with sorafenib, which is an okay drug. There's no drug, nicotinamide, sorafenib, and together they're better than either one of those two. So there's a synergistic effect here. And this drug is just sitting there in public data, a worm drug, waiting for you. Now, I want to make this point here. I'm not up here talking to show you how smart I am. I'm up here talking to show how smart you can be. I have no fancy sequencer in my lab. I have no mass spectrometer. Hell, I don't even have my own computers anymore. We run them on Amazon, right? It's not about the equipment. The data is just there. You can get to all of the same data I got to here for this. That's the whole point here. And we could do many more of these. And what do you do in Silicon Valley? You start a company on this. That's NewMedi. Raised more than $7-8 million now. They have these deals with Allergan, your neighbor, right? So this little team of 4 full-time people are developing the next psoriasis drug for Allergan, right? It was going to be the behemoth, right? If they merged with Pfizer, it would be the largest pharmaceutical company in the world. Allergan by itself is huge. So I call these garage biotechs. Garage biotechs. Because we revere garages in Silicon Valley, right? They're national monuments where HP and Apple started, right? You know. But I think the next Genentech, the next Amgen can start in a garage. Because when you go home to your garage, you got 2 million samples and every mouse model you can think of available on a credit card here, right? That's why I think about garage biotechs. And I don't mean making meth in your garage, no. I mean cancer drugs and diabetes drugs, right? Not Breaking Bad. So this little team deals with Allergan, Astellas, and now is working on idiopathic pulmonary fibrosis here. All right, where are we going next? What's the next big open data? What half my lab is invested in? What I think is going to be the next big thing to skate to where the puck is going? Clinical trials data. What's a clinical trial? The most expensive experiment in the world. That's why it's costing us billions to do this drug development. Half of them fail, you know that? When they fail, we don't even write papers about it. Never mind releasing the data. Oh boy, is that going to change. EMEA, European FDA, right, is requiring raw clinical trials data. We run this repository. FDA might get there. We run this for NIAID, part of NIH. This is one that we run where we released more than 100 raw clinical trials data to the public. And we're learning how to do meta-trials and meta-analyses and patient subsetting, comparative effectiveness. I think there's a lot you can do with clinical trials there. This is where I'm skating to, because I think the FDA is going to eventually require all that raw data to be released to the public. De-identified, we're not going to know who these people are, but can you see, maybe the drug worked, even though you say it failed, maybe it worked in a few people, and look for markers and all of that. So we have a whole bunch of stories here that I won't spend time talking about. Let's get out of molecular for a second, just talk about wearables for a second here. How many of you, show of hands, have a wearable or an Apple Watch, heart rate? Yeah, so it's usually about a 10% audience, which it is here. I used to laugh at these people, right? You know, quantified selfers, right? They measure everything that comes in and out of their body, you know? Measuring things that go into your body is tractable. Measuring things that come out of your body sounds gross to me. Why would you do this voluntarily, right? So I used to laugh at these quantified selfers until December of 2012. December 2012. Came back from a trip to Hawaii. I stepped on a scale. I was 247 pounds. I thought to myself, when I hit 250, they can measure me in tons, right? That's a quarter of a metric ton, right? So what the hell happened to me here? I'm driving everywhere. I used to walk. Bought everything Fitbit makes. Otherwise, I'm not connected to the company, just as a customer. Lost 50 pounds in 2.5 years, okay? If you don't believe me, look at my TED Talk. You'll see what I used to look like here, right? So I learned a lot. I write down everything that I eat, including all these things. I just got to write it up. I've been writing it down for like 5 years. I weigh myself every morning and all of that, right? And what's amazing is like this stuff works if you're an early adopter, if you're kind of really kind of motivated about it. And every one of my friends on Facebook knows that I've lost the weight, okay? So it's a whole social network thing. But then I went to my doctor, and it's been in every newspaper. This is part of Sutter Health. They spent $1 billion on Epic, but my doctor was the last one to know I lost 50 pounds, right? Because their $1 billion system doesn't talk to my $40 gadget. Can you smell the opportunity here, right? Right? And we in medicine have got to get our act together because there are hundreds of Silicon Valley companies that want to own the patient experience, especially the healthy ones. If we don't start to connect to patients between encounters, we're going to lose them to people, to companies that are all over this space. And we can't just keep saying Epic doesn't support it as an excuse, okay? There are a bunch of universities that are doing better. Stanford, UCSF, USC is all over this too with diabetes monitoring, but we all got to get closer to connected patients in between encounters. I'll talk about a couple of cool things that we do in California. As usual, California is way ahead of the curve. If you didn't know, Jerry Brown talked about precision medicine in the State of the State address a whole year before President Obama. Did you know this? Right, a whole year, Jerry Brown actually talked about precision medicine in the State of the State address. Now, I'll be the first to admit, I didn't even know he gives a State of the State address, but in January he does. I didn't know this, right? And he launched, so far he's spent $13 million over 2 years, where we're just funding, I'm the principal investigator for this, we're funding all sorts of interesting precision medicine projects around the state. These are projects between academics, so UC and non-UC academics, small and large, tech and biotech, all coming together. You got kids with cancer, you got sequencing for infectious diseases, we've got a whole bunch of these projects launched, so I'm really proud to represent this effort for the state of California. And now I'm at UCSF. So I'm running this Institute for Computational Health Sciences. We've been there for about 2 years. We have 38 faculty with all of these kind of distinctions. They were all scattered, and now they're all coming together in one academic home. We're slumming it out in this brand new building until they build our brand new building right across the street here. So Illumina is here. Our brand new hospital is here. So women, children, and cancer are in this part of Mission Bay. And Med-Surg and everything else is on the other part of on our Parnassus campus, if you've been there before. The ballpark is here, and this is where the Warriors Stadium is going to be, literally right across the street there. So everything's going to be in Mission Bay. If you haven't been to Mission Bay, you should go there. It's amazing the development that's been going on there. And that's where our new building is going to be right. But for now, we're in this building here. And the idea is, what are we going to do here, right? So we have an academic home. We got research development. Of course, we keep spinning out companies, develop new educational plans, and build and use our new data assets for precision medicine. So I'm going to end with that concept. What are these new data assets? Why did I leave 10 years of Stanford to move to UCSF? Why would I move? Now, of course, I got this building, I got the recruit faculty and all that. But for an amazing reason, they put me in charge of building a central data warehouse for all the clinical data for the entire University of California health system. So not just UCSF, but UCLA, UC Irvine, UC Davis, and UC San Diego. And the idea here is just launch it. It's going to run by itself, but they need someone to just at least get this whole effort launched. And what I mean is putting all the data together for 15 million patients. Did you know 15 million patients get some care in the University of California? That's 4% of the US population we care for. It's a crazy number. And now we have all the raw electronic health record data. Every dose, every vital sign, every side effect, every lab test result for 15 million patients. And that's that final challenge. How do we learn what's working and not working in medicine here? We're building off of something called UC Rex. Now we're building a central database instead of a federated database for people who like that kind of lingo. And I think I even have boxes here. I wouldn't be an IT person if it didn't show some slide like this. UC Davis, LA, Irvine, San Diego, and UCSF all going to one place. Riverside has a medical school, but they don't have a medical system yet, so they'll be the 6th one eventually, and just a big data, clinical data warehouse in the middle. Why are we doing this? Because we have a business reason to do this. There's a national narrative that it's so hard to do this. It's so hard to harmonize electronic health record data. It's amazing how much easier it gets when there's a business reason to do it. The University of California partnered last year with United Healthcare, the largest payer in the United States, and we've announced that we want to eventually build a single giant accountable care organization for the state of California. Now, admit they might not be called accountable care organizations. There'll be some other new name for them as healthcare evolves over the next 3.5 years. We'll see. But you get the idea. So we're going to act as UC Health. That's the goal here, business reason to share data, look for variation in care. We already used this to become part of the precision medicine initiative. I won't spend time talking about what are we going to do with all this data? Well, I actually think there are a lot of things we could do. AI researchers of the future can start to predict what's happening, and app designers, maybe we can inspire entrepreneurs with this data. Community activists can start to look at the clusters of diseases. Cancer genomicists should look at every cancer case that's out there. There's 10 bullets here for all 10 campuses of the University of California. I'd like to have this going to the entire, not just the medical ones. Everyone should have access to this. But in the last minute, what am I doing with this data? I'm building maps. Maps of death and disease in California. So what does that mean? Well, think about Google Maps. Google Maps takes you to pleasant destinations. I think about how people die in the state of California, okay? Unpleasant destinations. Here's an example of a map that from real California data, patients show up with alcoholism. Each arrow here is a year. So a year later, we see patients showing up with chronic liver disease or cirrhosis. And a year after that, they can show up with liver abscess. Or you could go this way. So you can go to the right, or you can go straight down in 1 year. And the squares mean you die of those diseases. So we don't see patients dying of alcoholism, but they come in a year or 2 years or a year, and they die of these cirrhosis or abscess. Do you understand this concept of a map? You see the arrows there. Hannah and Jay did this work for me. The maps get kind of interesting. Woo, 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 go back. Here, here's a more interesting map. Patients show up with heart attack. You can die right away. A whole bunch end up with heart failure. That means the heart isn't strong enough to pump the blood that the body needs. You can die of that. Because the heart is weak, the fluid backs up into the lungs. You can end up with lung disease. And then a whole bunch of patients die of sepsis. What's sepsis? That's a blood infection. Now, I kind of didn't know this, that you show up with the heart attack, but you're dying 3 years later of the infection, not the heart. So it's actually not the heart that's killing you. It's actually the infection that's killing you. Kind of didn't know that. We don't even really screen for that. We keep looking at the heart, but we maybe should be looking for these infections. If you don't take the north route, you go take the south route. Instead of the lungs, your kidneys get killed, and then you go end up in sepsis there. So I think about maps like this, right? And where do patients actually go? Not just encounter to encounter. What do we think is going to happen next? And so my goal when we open that brand new building in 2019 in Mission Bay is we're going to have a big conference room with a wall of monitors. And we're going to have all the maps of how you die in California on the wall there, right? Now, I love Google Maps. There's still one thing Google Maps cannot do. Google Maps cannot show you all the cars on the map. I know it shows you the traffic, okay? It doesn't show you each and every car on the map. How could it do that, right? It would be ridiculous to think about Google Maps doing that. So when we open our new building in 2019 and have our wall of maps, we're literally going to be able to show every one of our 15 million patients. This is real California data. This is a prototype. How patients actually move from disease to disease to disease to death. The age is getting older. The colors are getting brighter. A whole bunch of them are going to end up in sepsis in a moment and then die. There they go. And we can start to predict what's going to happen the next 90 days. And what's going to happen the next year. And what are we going to do about it? And this, to me, is going to be the new definition of an accountable care organization. One that knows how to account for the care of its 15 million patients. And this is what we're building in the University of California and nowhere else. So you should be proud to be affiliated with the University of California. This is what we can do. So what is big data and biomedicine in the end? Is it algorithms, programmers, databases, Hadoop, high-performance computers, mobile? It's kind of about all these things, about none of those things. It's about predicting diseases before they strike, explaining rare diseases that defy experts, finding drugs for disease that lack attention, not enough pharma companies on the planet, making sure we write safe, cost-effective thing for patients, an amazing platform for biomedical innovation and entrepreneurship. All this to sum up with one word, hope. Patients, families, and doctors are counting on this way of coming up with new products and services. All this data is sitting there waiting for you. It's up to you to bring your expertise and deliver this hope to patients because the data is already there. I thank an enormous number of people at the University of California that helped build that data warehouse. I used to color code them by campus. I no longer do that. We are all one giant campus, just different zip codes. 50 people working on that data warehouse. Long list of collaborators that help with all the basic science to get the work done for us. I thank UCSF for endowment support. My lab has 20 NIH grants from these 10 institutes of NIH. These 6 give me more. These 4 give me less, but I still love them. March of Dimes, JDRF, HPH is my stem cell money. A lot of disease-specific foundations want us to work on their diseases. I always thank my admin and tech staff. I never get a grant or paper out the door without them. Thank Zach Kohane for being my mentor and friend for life. Sam McKee for recruiting me to UCSF. I always thank my daughter, Kimmy, because it's very important. She's 14 years old. She's a science geek, and she's happy being a geek. We need to keep more of these kids interested in biomedicine. As middle schoolers and high schoolers, they're turned off by dissecting frogs and formaldehyde. Biology and medicine can be very digital. We got to show them this cool aspect of it too. And I always thank my wife, Tharani Deshpande, a molecular biologist and biochemist. She reads every major paper grant comes out of my lab, collaborates with my lab a lot, starts companies with me now, and lets me go all over the world to give talks like this. Thank you very much. Went a little bit over, but I think maybe we still have some time for some questions here. Don't be shy. And I'll repeat the question. Go ahead.
Yeah, you had that one slide that showed one company spends $4 billion per drug.
Yeah.
6 billion, 8 billion. The part about that slide that is kind of hidden is there's probably 100,000 drugs that they prove that don't work.
That's right. That's the cost.
Is there any sharing between these companies? Like, hey, we already did that.
No. That's another really side effect of the problem right now and why clinical trial sharing is going to be helpful. You can have companies launch an expensive clinical trial. They might get a lot of internal data. But when it fails, sometimes things fail and you can see why they're going to fail. But for complex things like cellular therapies, for example, for cancer, big trials failing, they pull the plug and nobody knows why they failed, right? It's kept internal. And then we're all doomed to repeat those experiments again, right? Because we're not learning from the biology. So sharing, I think there are going to be a lot of mandates to share data because, first of all, it's more reasonable. Now, there are pre-competitive efforts, what they call pre-competitive, where more science can get done in a sharing level, so it does happen. They also work with NIH to get more of that done. But in general, the expensive end of this, the trial end of it, those fail or not, it's hard to get that kind of data today, especially when they fail.
There's a business opportunity there. Give me your data and I'll...
Yeah, everyone tries this, right? So the problem is, the minute those trials fail, or even when they're close to failing, teams want to leave right away, right? You don't want to be part of that sinking ship. So, in fact, it then becomes taboo to even talk about these trials in the companies, right? And then the half-life of that knowledge, that expertise drops rapidly within weeks. And then people don't even know where were the files, where's the data. So if you do this, you got to do it in real time as it's failing. Come back a year later, of course, that would be a great place to salvage drugs, reposition drugs, all that. We've done a lot of thinking. You end up, the finances are reversed. You end up having to pay millions to those companies to resurrect the files because they're just lost to the winds the minute that these things fail. So it's a weird scenario. I don't know. Again, there are probably pharma people in the audience here, so I'm not going to be too critical. I consult for them all. You saw my second slide here. But I don't know why shareholders tolerate this, to be honest with you, right? Because the shareholders of these companies, you poured millions, literally millions, if not billions, into the drug, and then it goes to zero value. You'd think you'd want to still regain some value here, right? So I think it would be that aspect that would put some pressure on companies, if anything. But it's rare. A lot of companies do have repositioning efforts, but they're really ad hoc right now. Nothing consistent. Yeah, go ahead, and then we'll go to you. You first, sure. No, my pleasure.
Early on, you talked about the fact that there are other components to patient preferences.
That's right.
And things of that sort. But most of your talk really focused on clinical.
That's right.
So can you talk a little bit about what your plans are in that area?
Yeah, so patient-generated data is a huge new opportunity here, right? And in general, we try to estimate patient preferences during the clinical encounter. So a doctor might ask you, these are the choices. Do you want to go for prostate surgery, or do you want to watch the cancer? Things like that. You get an idea during the encounter. But they're collected in a very ad hoc way. So there are companies trying to get digital aspects, trying to collect patient preferences that way. In trials, it's also still early days right now. In general, though, I think it's really done in an ad hoc way. We have no real way, we don't capture any of that information except to see what was done in the end. Now, the doctors themselves don't get a lot of advice from computers either. That's another part of the problem. So we have Epic and or Allscripts or Cerner or any of these electronic health record systems, but in general, the doctors use them to document their notes. It's super rare to find a doctor that uses those systems before the patient comes in. And they definitely don't use it a day before the patient comes to get advice of what to do, right? So the advice-giving, decision support is really in its infancy. Patient preferences as a component, key component of that is really in its infancy still, to capture in a digital way right now. You had a question back there.
I'm kind of curious for your opinion. So in the end of your slide and your prototype, you're showing that a lot of these people are dying of sepsis.
That's right.
But in big pharma and other things, there's no sepsis drug on the market because we call it prayer.
Oh yeah, of course.
So my question is, in your opinion, do you think that you can change the way they invest in drugs, let's say sepsis, if you can prove with all these big data decision points in order to treat?
Yeah, it's great about sepsis and what to do with it, and could we actually influence? I mean, so the raw numbers are there. So people know, a lot of people end up sepsis. It's a 50% mortality rate. The minute you carry that diagnosis of sepsis, you've got a 50-50 chance of living in the United States. So we do use that diagnosis a lot. It covers more than just bacterial infections or viral infections. Multi-organ system failure usually falls under that and things like that. And it has been a graveyard of drugs, but we're still going to have to keep trying to come up with drugs for that. Earlier use of antibiotics is there. There are a lot of people trying to come up with informatics methods or earlier diagnostics. If you can gain 6 hours or 12 hours on that diagnosis, maybe you can prevent some of the deaths. So people are trying that aspect. Earlier therapies, some of the therapies we already have. People looking at signal processing to try to look at monitor traces and stuff to get to that diagnosis earlier. But it's a tough one. I mean, it's a variable diagnosis. It's not really clear. Can you point to a patient and say, you didn't have sepsis here and you have sepsis now? You know, all those things that make machine learning hard. There are a lot of, it's a challenging one. But we do use that diagnosis a lot, and patients end up having a high mortality from that. So we do have antibiotics, but we probably could use earlier diagnostics there. Sure.
Okay, then we want to thank
Thank you very much.